Good morning. Welcome to Freeslate's webinar, A Novel Automated System for Buffer Exchange and Concentration of Biopharmaceuticals. My name is Susan Darling, and I'm the Director of Product Marketing at Freeslate, and I'll be your host today. This morning, we're going to go through an introduction of Freeslate's automated buffer exchange system and how it applies to protein formulation. And we'll look at data comparing this system to traditional techniques such as dialysis and Amicon. We are pleased to have Dr. Russell Burge from Freeslate join us today to give this talk. And at the end of this webinar, we'll finish up with a brief Q&A session, and that will conclude the webinar. Before we begin, let me invite you to submit questions using the chat window on the lower left-hand side of your console. Any questions that we do not get to will be answered privately in a follow-up email, so please feel free to submit questions throughout the presentation. As I mentioned, our speaker today is Dr. Russell Burge. Russ earned a PhD in molecular biology and biochemistry from the Scripps Research Institute in La Jolla, California. He worked on characterization of aptamers as part of his postdoctoral training at the University of Colorado in Boulder. Russ also worked as a formulation and analytical development scientist at KBI Biopharma, where he contributed to numerous biopharmaceutical development projects. Currently at Freeslate, Russ is an application scientist where he designs and performs demonstrations of our systems used in research and development of pharmaceuticals. He additionally contributes to Freeslate with marketing materials and development of new technologies and products. The title of the presentation today is A Novel Automated System for Buffer Exchange and Concentration of Biopharmaceuticals. And now I'll hand it over to Russ.
Thank you. Today I'll be talking about traditional versus high throughput formulation development. So what's being done currently in formulation development labs versus some of the technologies and workflows that we have been developing at Freeslate. Specifically, I'll be introducing our automated buffer exchange system, which is a culmination of research and development that we've done at Freeslate and collaborations that we've performed with pharmaceutical and biotechnology companies. Specifically, I'll be talking about a few case studies where we used the automated buffer exchange system to understand the applications of the system for therapeutic proteins. So the first case study will be comparing the Freeslate method to Amicon devices and other what we would consider traditional formulation development methods of buffer exchange. And then we have an accelerated stability study of material, specifically a therapeutic MAB, that was buffer exchanged by both the Freeslate system and traditional methods. And then I'll be wrapping up with a summary and conclusions. Traditional formulation development requires buffer exchange to be performed in order for formulators to understand the product that they are developing and understand how it may be turned into a drug product and manufacturability. So in this particular case, we see that the traditional procedures that are used at the earlier stages and at smaller scales are oftentimes time-consuming. They require quite a bit of manual labor, and they also require quite a bit of material, both drug substance and dia volumes and other volumes that are needed for buffer exchange. So because of these challenges, formulators are oftentimes limited in the number of formulations they can truly evaluate, from pre-formulation all the way to even late stage. And these limitations can lead to gaps in the product knowledge. Specifically, how is the formulated drug substance behaving? What is its robustness? How may those properties affect drug product and maybe even manufacturability? And so here's just some examples of some of those traditional methods. So Amicon devices here on the bottom left, and then on the bottom right, something like a dialysis system. So to go into more detail about these what we're calling current buffer exchange methods, we're highlighting three here. There's dialysis, ultrafiltration, and desalting columns. Notably, there's also TFF, which is oftentimes used at larger scales and at later stages when there's more material. But at these smaller scales, these three methods are the most prominent. Now, their advantages are all pretty similar. They're relatively easy to use. They're relatively gentle on the protein, and they all have relatively high protein recoveries. Where they have some separation is their disadvantages. So they have now, in the case of dialysis, it takes quite a bit of time for the material to be processed, and it requires quite a few manual steps. So at least you're looking at dozens of hours to maybe a day to perform dialysis. It requires large volumes for exchange, meaning the buffer itself, and sometimes it can even require quite a bit of material. It's rare that buffer concentration is adjusted during dialysis, so this is the protein cannot be up-concentrated. In the case of ultrafiltration, the one notable disadvantage or feature of this method is that during filtration, because this is a dead-end filtration, there can be a layer of protein that will amass on the membrane, and that can slow down the filtration. So this is what we're referring to as polarization on the membrane, so a local concentration of protein. And then with the desalting columns, we have the issue that the protein will be diluted, and sometimes that dilution factor is significant. And so the formulators have to adjust the concentration typically after the processing. And what's also important to note is that desalting columns might not necessarily be compatible with all formulation types due to how much salt might be in the initial and final formulations. So with high throughput formulation development, we're trying to address some of these challenges. So for instance, we are leveraging the technology that we have here at Freeslate, both automation and software and data management, to create a system that can perform high throughput and fully automated buffer exchange at smaller scale, which will allow formulators to rapidly screen formulation space, as we're calling it, and they can get a better understanding of their drug products and formulated drug substance earlier on and throughout the project timeline. And when we go to these smaller scales, the idea is the formulators can use less material. So I'll be talking about how we can use something as little as a few micrograms of material in this automated system. And because it's fully automated, once the process is going, the formulator and technicians and scientists don't have to have any manual intervention with the system. So specifically, what we're using then to draw your focus here to this consumable plate, this is a 96-well consumable, commercially available, inexpensive molecular weight cutoff plate. And the idea here is that it has a 10 kD cutoff membrane, and this is the system that we decided to work with because it's microscale and it's very amenable to automation. But the idea is that we can actually take some of that material and then move it on to devices such as serum vials and prefilled syringes later. And the idea is that when we apply this system at multiple stages or various stages of drug development, it enables the formulators and their colleagues to get a large amount of information that will help them understand the robustness of their formulations, and it will accelerate development at not just the formulation scale, but also drug product and manufacturing scale. And as I alluded to earlier, because we have the ability to manage data, even though we're going to create a lot of information, we also manage that data very well so that the scientists can leverage it and understand it and share it with their colleagues efficiently. Okay, so let's talk a little bit about the automation of the buffer exchange process. So we spent quite a bit of time and effort here at Freeslate with our collaborators and also independently to understand what was the best way to automate buffer exchange. And where we ended up was using a pressure-based ultrafiltration diafiltration method. As you saw earlier, I sort of alluded to the filtration plate that we're using. But we have a process that differs a little bit from how those plates are normally used. So in this case, we're using 60 psi nitrogen. This is clean, dry nitrogen, which is applied across the filter plate to create the ultrafiltration step. We can also apply gentle orbital mixing to prevent that polarization at the membrane I mentioned earlier. Now, we can do this, but we don't have to do this. This is up to the user to add orbital shaking if they'd like or not. And then at the end, there's an additional optional step, which is a concentration step. And in fact, if you want to, you can use the system specifically to concentrate as well. But the system can buffer exchange and then concentrate. So the workflow is as such. We start with adding the protein to that 96-well filter plate that we saw a moment ago. And you can see that we know how much we added with the liquid handler, but we actually use a volume checking device then in step 2 to verify the volume in every single well. And then we use the pressure-based filtration, as I mentioned earlier, to provide that ultrafiltration step, which then removes some of buffer 1 and keeps some of the protein in solution. And then after that filtration step, we measure the volume again using the volume checking device. Now we have this delta volume. Because the system is all integrated and automated, that delta volume information is sent to the liquid handling device, and then that new buffer is added to the original volume or some specified volume that the user may want. And now this is how we're replacing buffer 1 with buffer 2 while maintaining a certain solubility limit of the protein. Now we repeat steps 3 through 4 multiple times until the buffer 1 is exchanged out. Now, we recommend about 99% buffer exchange, but the user can actually set this to what fits them best for their application. And as I mentioned before, step 6 is this optional concentration step, which is very simple. It's basically step 3. We're pressurizing and filtering, removing the buffer, but now we're not adding any diavolumes back. And this will allow us to adjust the concentration of the protein. How do we do this? So as I mentioned, we've spent quite a bit of time and we've devoted a lot of research and collaborations to develop the system and to validate it for the application specifically of buffer exchange for biopharmaceuticals and biotherapeutic proteins. So here's an example of a system that has fully automated buffer preparation capabilities. It can buffer exchange, and then it can take those materials that have been buffer exchanged, it can stress those samples, it can create analytical sample plates for, let's say, DLS or UPLC, and the system can also perform analyses such as viscometry, pH, and so forth. So how does the system work? So on the right side, we have our CM3 automation system. This has a multitude of different capabilities. It has the ability for liquid and solid dispensing, so you can start with something as easy as Wi-Fi and some powdered materials. You can create those formulations that you want to buffer exchange into. The system will then interact with, on the bottom left side here, you can see in silver, this is the pressurized chamber that performs the UF step. There's a blotting station which removes liquid off the bottom of the filter plate. And then at the bottom left here is the volume measuring device, which is non-invasive. It uses ultrasonic technology to measure very precisely the volume in every single well. So to go into more detail about the workflow, as I mentioned before, there's a liquid handler on the right side of this automated system, which adds about 450 microliters of protein to each well. That's what we recommend, but there's obviously you can add a little bit less if you'd like, but it's in the hundreds of microliters of material for the protein for each well. Then the system automatically moves the filter plate over to the volume checking device. We get this very precise volume measurement to verify that what we've added to that filter plate is correct, and we have additional information. Again, it is non-invasive, and it does not actually contact the material. It uses ultrasonic waves, so there's no contamination here. Then the plate is moved over to the filtration, the pressurization stage where filtration occurs. And here, this is where we then apply the pressure across the membrane. And again, we can filter, but do not have to. We can mix, but we do not have to mix during the filtration. The filtrate does go to a shared waste, and then the plate is moved back to the volume checking device to verify how much volume was removed from each well. That delta volume information, as I mentioned before, is communicated to the liquid handler, and the liquid handler adds back the precise amount of volume to fill back the wells so that you can replace the initial buffer with a secondary buffer. So in this case, or formulation. So in this case, we have formulations A through G as an example. And in this case, we're each row corresponds to a specific formulation. This is just an example workflow. And then these steps here are repeated until 99% is exchanged. Now again, that's user specified, and this format does not have to be like this. This is just an example. And for most of the proteins that we've studied so far, even at higher concentrations, we can get this process down to about 2 to 10 hours, meaning 99% exchange is achieved, and the plate can then be moved for sterile filtration, and the material can be moved on to other applications. So going into common formats, we now have here are some examples of what are most commonly used and what those applications are. So we have the 96 different unique formulations. We have 8 unique formulations, which I showed earlier, and then there's also the possibility of just doing one formulation. These aren't the only formats available. There are other options in between these. These are sort of the extremes and the most commonly used. So in the case of the 96 unique formulations, this is ideal for earlier formulation development, possibly even pre-formulation development, where you're looking at screening of the buffers. You want to look at robustness, and maybe here you're still looking at pHs and salt concentration and very early excipient screens. Then you can take some of the information that you've gleaned from that earlier study, and you can move on to something more like 8 unique formulations, where here maybe you're trying to optimize the pH or the excipient concentrations. You can also perform solubility studies where each one of these rows may correspond to a slightly different concentration of the protein, or maybe adding different excipients to stabilize a protein at a certain concentration. And then we can also use this material to put on stability studies. So one of the applications we can do is take the material from these 96 well plates. We can pool all of the material from one row, and we can use that material to fill drug devices such as serum vials or prefilled syringes or small glass vials and put those on stability. And then in the extreme case, we can make one unique formulation. This is applicable for larger scale preparations, and maybe this is something where you're looking at very specific studies like a DOE or an excipient optimization for the protein, and maybe you want to take this material and create more prefilled syringes or serum vials for your stability studies. So now I'm going to talk a little bit about some of the case studies that we've done here to show the applicability of the system for therapeutic proteins, not just MABs, but other therapeutic proteins as well. And then we're going to compare the performance of the Freeslate system to what we're calling traditional buffer exchange techniques. So study one is quite straightforward. We paired and worked with several biopharmaceutical companies and biotechnology companies to get therapeutic proteins. In this case, we have MABs 1 through 4, so we were able to get multiple MABs from multiple drug developers. We also have an enzyme and a fusion protein that we worked with to use this material to understand the performance of our system. So we took these solutions and then we split them into 2 studies. We buffer exchanged them with the Freeslate automated system, and we buffer exchanged them with Amicon. And then we looked at various bits of information such as recovery, protein concentration, pH accuracy, and protein aggregation. So first and foremost, we have protein recovery. Obviously, it's very important. Whatever material you put into the system, you want to make sure that you're recovering. So we did a study where we looked at the mass recovery of all of these molecules. What's important to note is that we have 6 different molecules. They represent 3 different types of therapeutic protein at various protein concentrations. So we're ranging from 11 to 71 mg per mil protein that's being pulled out of the system. And what you can see here is when we compare the Freeslate system to the Amicon system, we're getting at least 91% of mass recovery on the Freeslate system, which is relatively comparable to what we were seeing with the Amicon system or the Amicon device. When we look at our accuracy of our final formulation, when we want to talk about protein concentration, we're able to target the protein concentration of the final formulation very accurately. So what this plot is showing is the protein concentration yield as a percentage of target. So if you're at 100%, that means you have exactly the targeted protein concentration of your final formulation that you intended. If you have 90%, it means you're off by, you have less protein concentration than you intended. And where our system has an advantage is because we're able to measure very accurately the volumes, and because the volumes are directly proportional to the concentration of the protein in every well, we can very, very accurately target a protein concentration. And in the case of Amicon, which is shown in gray, oftentimes there is an underestimation of the concentration in the final formulation, which is simply due to the fact that with the Amicon device, it has to be manually there has to be a manual visual inspection of the Amicon device, and maybe you can use an air displacement pipetter, but it's not going to be as accurate as this ultrasonic wave volume detection system that we use. The automated buffer exchange system can target pH values as well. So in this case, we have another set of concentrations for the same 6 proteins that were shown before. We have a pH that the protein was initially formulated in. Then we have a target pH in the next column. And then the preceding 2 columns are the post-buffer exchange pH for the Freeslate system and Amicon device. And then on the far right here, when you look at the delta pHs for both devices, you can see that the deviation from the target is very small. In most cases, it's less than 0.04 pH units. And the only deviation from that is with this MAB4, which is at 50 mg per mil MAB concentration. And there we see for both of the methods slightly higher deviation, but still within 0.09 pH units. And this is likely due to the buffering capacity of the protein at the higher concentration. So we also wanted to look at SEC of material that was processed by the system. This was very clear. We had a 40 mg per mil MAB. This is a representative MAB from the 4 that we were talking about before. In this case, it's a chromatogram overlay of the unprocessed stock, material that was buffer exchanged with dialysis, material that was buffer exchanged with an Amicon device, and we have 2 replicates of the Freeslate buffer exchange system. And what you can see here very clearly is that they overlap almost perfectly. So this is suggesting that right after buffer exchange, there's no aggregation that's occurring due to buffer exchange. We also have the SEC results after buffer exchange for all 6 of the therapeutic proteins. So previously I showed a representative chromatogram, and here's some of the data for the 6 therapeutic proteins we were looking at. So on the left side, we have the monomer percent area. So this is simply looking at the stock versus the material that was buffer exchanged with Freeslate and Amicon devices. And what you can see here very clearly is that there's no significant difference in the monomer content of any of these proteins when you compare the stock, the Freeslate, and the Amicon devices. They're all very similar within the error of each other. This is suggesting that the buffer exchange for both of these systems is not affecting the aggregation state of the protein. Likewise, we looked at the aggregate itself. So here we're looking on the left side, the aggregate peaks percent area. And again, when we compare the stock, Freeslate, and Amicon results, we see that they're all very close and within error of each other. This is again suggesting that the aggregation is not, any aggregate that's in these proteins is not being affected by the buffer exchange significantly. So moving on to our case study 2, we wanted to ask the question, okay, we now understand right after buffer exchange, it's clear that there are no significant differences in the aggregation state. But we wanted to understand what happens at a longer period after buffer exchange. So we decided to perform an accelerated stability study where we took material that was buffer exchanged and we put it on stability and accelerated stability at 40 degrees C for a multitude of weeks. And then we took material from the incubators, and then we performed SEC, DLS, and CEX to understand the stability of the protein. So study 2 is very simple. We took a representative MAB, MAB1, from the first study, and we took the material and we buffer exchanged it with the Freeslate automated system and with Amicon device. And then we took that material and, as I mentioned, put it on 40 degrees C. I'll be highlighting the results at 4 weeks and 9 weeks, but we had several other polls that we analyzed. And in this case, we really focused on SEC, DLS, and CEX because of the information that we can get about the physical and chemical stability of the protein as a function of its processing and as a function of its incubation. So here, this is MAB1 after 9 weeks and also T0 for reference. But if we focus on the 9-week chromatogram, we see is that the stock is in blue, the Freeslate and Amicon are in red and green, and what's very clear here is that the two buffer exchange materials overlay almost perfectly, and this is suggesting that the protein that was buffer exchanged with either of these methods is as good as, if not better than, what the stock was in, or how the stock was performing. Another way of looking at this is monitoring the aggregation state via the main peak percent area of the SEC over time. So as you can see here, we again have Freeslate in red, Amicon green, and a stock in blue. And as we follow this from the course of several weeks, we can see that the percent of the aggregate species does not significantly change from the stock to the buffer exchange materials. We also looked at the DLS results for T0 and at a later time point. And this is important to see if whether or not the monomer is changing either in its radius or PD. And additionally, we're looking for any other small peaks that might be arising due to buffer exchange. And what we see very clearly is that the DLS data shows that there are no changes at T0 from the material that was buffer exchanged when we compare that to the unprocessed control on the left. Put this material on the stability, and at 4 weeks we were looking at the stock and comparing that to the MAB1 after it was buffer exchanged with the automated Freeslate system. And what we see is that there were no significant changes between the buffer exchange material to T0. At the MAB stock, it had some small differences there, but this may be attributable to the slight difference between the initial buffer and the secondary buffer in which the material was buffer exchanged. So essentially, there's no difference in the MAB1 after buffer exchange and after incubation at 40 degrees C at 4 weeks. We have a summary of this data, and this is also showing that the stock, the unprocessed stock, has a slightly larger radius compared to both buffer exchange material. Again, this may be due to the fact that the stock is in a slightly different buffer than what we buffer exchanged into. And then we can see that the PDs were quite good, very low PD percentages. And this is also showing that between T0 and 4 weeks, we're not seeing any significant changes or just a very slight change in the percent PD. But this would be suggesting that the protein is staying quite stable after buffer exchange and incubation. We also looked at CEX results for MAB1 at 40 degrees C. So here we have a chromatogram which is slightly offset, but you can see that that the chromatograms are extremely similar between what was buffer exchanged with the Amicon and with the Freeslate system. And then when we look at this data over the time course, we can see that at T0, the percent main peak, which is specifically sort of cut in this area, this would be the main peak, and anything outside of that would be the acidic and basic peaks. We can see that the percent main peak is very similar across the stock, the Amicon, and the Freeslate. And as time goes on, the story is basically they're very similar percent peak area, main peak areas, all the way into 9 weeks. There are some slight differences, but overall the percent main peak is at least as prolific in the Freeslate and Amicon as it is in the stock after 9 weeks at accelerated stability. So with this information, I'd like to make some summary and conclusions about what the work that we've done specifically here at Freeslate with this material that was contributed to us by pharmaceutical and biotechnology companies. The first thing is very clear. We are able to hit very accurate pH values and protein concentrations. We're also able to have high protein recovery. We can have the system can recover the protein for you out of the wells, or if you choose, you can also manually pipette the material. I wouldn't recommend that, but if that's something special for you, it doesn't have to be automatic, but we can automate this protein recovery into other vials or microtiter plates. And again, we have excellent protein recovery values for that. We're able to achieve this greater than or equal to 99% exchange in a very short amount of time for most protein concentrations and protein formulations. And we are also able to show the system does not affect the protein stability after processing. And we didn't just show this with MABs, we also have information with a fusion protein and an enzyme which suggests this. Now we've had additional studies from work we've done in collaboration with biopharma companies, which I can't show today, and we plan on publishing some of that material soon, but I can give you kind of an executive overview of what we've seen in those cases. So the collaborations with our biopharmaceutical partners demonstrated that the buffer exchange can be applied to a wide range of different formulations. Formulations that include protein concentrations lower than 1 mg per mil and higher than 100 mg per mil as the material that is initially placed in the filter plate. We've been able to work with material that has viscosities up to 46 centipoise, so relatively viscous material. And in the end, we've been able to have that optional up concentration step. We've used that step to get the protein concentration to at least 200 mg per mil for protein that's soluble and stable at 200 mg per mil. And that was after buffer exchange was completed. So this is showing some of the capabilities of the system. And when our collaborators had their own independent studies about the impact of the protein stability, they noticed that when they looked at a panoply of different analytical methods, they also saw no significant impact on the protein stability after buffer exchange. And one of the bits of feedback that we were receiving is that the system is providing significant time savings and increased productivity. And more importantly, this automation can do something that simply stated analysts and scientists cannot do in the lab by themselves. The automation can create these workflows that must be automated in order to be finished in an appropriate amount of time. So literally, this is impossible to do without automation. Summary and conclusions, this is building on what we've seen before. So now we're able to show with our in-house studies at Freeslate that we have comparable results to manual methods. So specifically, that's the Amicon device in dialysis. And we are also able to show that we can work with very small amounts of material. If you're at the sub-milligram scale, the sub-milligram per milliliter scale, and you want to work at the lower end of the volumes that the system can handle, you're looking at single-digit microgram quantities, so very, very low amounts of material can be used to get buffer exchange and some interesting formulation information. As we mentioned before, we can support multiple formats in the plate, and the automation can then take the material from those formats and move it rapidly into serum vials or other devices that, or put it into a secondary container that can then be moved on to other devices that are more applicable for your downstream workflows. Of course, when we have this breadth of information, this will improve the productivity and the product knowledge of the formulators during these steps in the project lifecycle. And the intention here is to have these systems in formulation development labs where we can use the system to very rapidly screen and optimize buffers and formulation components for many different formulations and many different molecules. Perhaps molecules that were not possible to be formulated before or had significant risks associated with them for developability and manufacturability. So with that, I'd like to thank you for attending. I'd also like to thank my colleagues here at Freeslate who perform many of these studies. And I'd also like to encourage you to reach out to my colleague Quincy Mehta, who can provide more information about the studies and products that I presented here. I'd also like to welcome any questions. Please submit your questions via the chat feature, and then we will address those now. Anything that's not addressed now, we'll move on to later. And with that, I will rest here with, I'll let you, I'll also invite you to come to freeslate.com. There is a technical resources and learning center site there which has previous webinars and other information that should be useful. So thank you again.
Thank you, Russ. As he mentioned, we will now start our Q&A session, and we have a number of questions, so we'll just get started with that. Anything we don't have time for, we will certainly, as Russ mentioned, follow up with you individually. So Russ, the first question we'll answer is, I'll have you answer, is the process under sterile conditions?
That's a very good question. So in our case here, the way we perform these studies is we have HEPA enclosures and HEPA filters, which will remove quite a bit of the particulate, and we can also use something like an IPA solution to sterilize or to some degree create an aseptic condition inside of our system. That would get you to what we would consider to be sort of aseptic conditions if we're running the HEPA filters, which makes somewhat like a laminar flow hood and doing the sort of pre-cleaning, pre-sterilization. I also know that these systems can be compatible with laminar, sort of a custom larger laminar flow hood as well, if truly, or shall we say, higher rating of aseptic and sterility is necessary for you.
Okay, great. Another question here is, because you're doing it's a 96 well plate and you're applying pressure to all the wells, is the volume controlled, or is there any difference in volume seen depending on different buffer viscosities? Do things sometimes filter faster or slower? Is there a way to control that, or how does the system manage the volume in 96 different wells?
Right. So that's a very good question. So individual wells are not really controlled with different pressures. The only thing you can do is, if you want to, you can close off a well if you'd like, and then it has less or no pressure. So other than that, if a well is open, it's going to be seeing a very similar pressure to all the other wells. Now, in the case of having formulations that have widely different viscosities, let's use the extreme case of 1 and 46 centipoise, or millipascal seconds, then yes, we do see some changes due to not just the viscosity, but also to oftentimes the drug product or formulated drug product concentration. And that's something that we have to address when we work out these workflows. So it's not suggested to try the processing the material with widely different viscosities on the same plate. But the system can handle that. It's just your bottleneck is going to be the slowest filtering material, and you're going to be adding far more buffer to your faster filtering material, because you don't want that to overconcentrate or dry out. So that is possible, but because of the slightly different filtration rates, it is something for the formulators and scientists to plan out, to sort of bin, if you will, the formulations that have similar flow rates.
Okay, great. And then another question here is, has there been any measurements to ensure small molecule excipient concentration is maintained in the buffer throughout the exchange process? For example, like amino acids, sugars, and pH adjustment buffers?
Yeah, that's a very good question, and we actually will have some of that information hopefully published here soon. I can say that I presented at the ACS meeting in March some results that showed that. And yes, we do have data which shows that not only are the excipient concentrations maintained, but we can actually hit those targets relatively similar to what we saw with the pH and the protein concentration. One thing to note is that certain types of excipients will have nonspecific interactions with the proteins, and that's something that we actually saw in that study as well. So the answer is yes, we can maintain those concentrations, and in addition to maintaining and targeting those concentrations, we also did a study where we looked at whether there was contamination, if there was any sign of cross-contamination of any of the small molecule excipient components, and we saw no cross-contamination. So even though these wells are very close to each other, they're only about 1 millimeter away from each other, the liquid handling device is so accurate that there's no amount of material that's being transferred or is being contaminated from one formulation to another.
Okay, and following up on that question, talking about cross-contamination with the excipients, are you generally using one protein per 96-well plate? Have you done multiple proteins in a 96-well plate, and have you seen cross-contamination, any risk of cross-contamination between the different proteins?
Right. So normally we work with one formulated drug substance per plate, meaning so it's one therapeutic protein molecule per plate, and then the variable is typically components of the excipients, whether it be buffer or pH or excipient types such as amino acids. So that's what we recommend. It's not to say that you can't use multiple proteins if they have similar flow rates and similar properties as far as how they work with the filters, but that's how we recommend to do things. And then maybe to follow up on this question and the last question, so yeah, we don't see cross-contamination, something we can easily control. That the liquid handling is a very advanced feature of the system, so there's many different ways to control the liquid so there's no splashing or anything like that would be a risk. We have very, very good control over that, whether the material is at low viscosity or even at quite high viscosity. That's pretty much a solved issue for us. The one thing I would say is, following back up on the previous question, is we've also done studies where we looked at buffer exchanging a component out of the initial buffer and verifying that component was gone after buffer exchange. We've done that with several components. Again, as I said, I presented that data, some of that data at the ACS meeting. We're not prepared to show it as part of this webinar right now, but it's certainly something that we can we plan on showing very shortly.
And Russ, I think it's fair to say the reason we can't show it is because it was done in collaboration with a biopharma company, and so it hasn't been. They haven't allowed us to. They haven't released that for us to publish yet, but that will be done shortly. So that actually followed up on another question we had: is have there been any measurements to show that the small molecule buffer excipients have indeed been removed after exchange?
Right, and that's something that's very important. So we showed that, and it's important to show that because there might be some concerns about, well, maybe the small molecule components are interacting with membranes or are sticking to the protein, et cetera. But what we've seen is, in spite of those possibilities, after 99% exchange, those excipients are removed using this system.
Okay, great. So what if a well stops filtering during buffer exchange or up concentration?
So we've developed some pretty sophisticated error handling to handle this what I would consider to be a rare case. So there are times when a well might stop, and when I say rare, it's literally 1 in 1,000 or 1 in 5,000 wells will have this issue. The system when it sees this, will actually know not to add any more dia volumes, because if it starts adding more volume to that well, the well will just start to overflow. So the system actually sees that as an error and knows how to handle that. The user then later can come back and say, oh, okay, that well had stopped filtering. How would I like to treat that material? Do I want to quarantine that material? Would I like to maybe pull that out and study it separately? Would I, in this case, do I even care? The protein is so important to me, I want to pull it back into other wells. These are all choices that the user can make depending on their particular case. So that's something that we've spent some time thinking about, and we've developed these sort of error handling techniques.
Okay. And let's see, let's take another one here. Are there any methods or procedures to use Freeslate to automate the production of a library of buffers and or formulations? For example, is there a good method to produce a library with different buffers at varying adjusted pHs?
Okay, interesting. Yes, there are actually many examples of this. So the current systems can mix stocks or other liquids together with powders at different concentrations, and that will create a library of different buffers rapidly with different pHs. The system also has an automated pH measurement system that can go through and verify the pHs, which is also very important. And then we can also just mix liquids together to create a variety of different formulations at different pHs. Now, that being said, there are also, as I mentioned, there are ways to check the pH with the system, and adjustment of the pHs should be done either with the components, or they can be done at a later stage. So yeah, the system has a multitude of different workflows to create different formulations and buffers rapidly.
Okay, great. The next question is about temperature control, and the question is, are you able to control the temperature within the pressure manifold?
Currently, the answer to this is we use room temperature to control the temperature of the pressure manifold. That's what we recommend, and currently that's what we have right now.
Okay, and have you seen any issues with doing the processing at room temperature in regards to protein?
No, we haven't seen any issues that are cropping up with proteins at room temperature. I would say that the question is important, and it is something that we've thought about. So either cooling the pressure chamber for molecules that are maybe a little bit more sensitive to higher temperatures, or heating the manifold so that materials that have higher viscosities or slower flow rates might have higher flow rates. So these are things that we've considered. We haven't done really any of the testing yet. It is a really good point, and it's something that we are thinking about, but currently do not support. And again, we don't have data on those other temperatures, so there's not really much for us to compare to. But when we look at room temperature, we're seeing that this is a highly applicable condition for these biotherapeutic proteins.
Great. Okay, just looks like we just have a couple more questions, and then we'll finish up. So what happens if the filter membrane loses its structural integrity, or if it leaks?
Right. So the way that these consumable items are made, each well has its own individual membrane. And again, just like the sort of stuck well that we talked about before, where the material stops filtering, these sort of blown out membranes, as we call them, membrane that sort of just pops or leaks quite quickly. This is very rare. This is a case where we're literally talking 1 in several thousand wells will have this issue. So in many cases, that material might be lost. But what happens is the system is able to continue to filter, and it will just treat that well as the system will look at that volume, and it will automatically know that the volume in that well is problematic, and will stop adding material, and will also provide immediate feedback to the user in the form of a red circle which says, okay, this system, this well is no longer functioning.
Okay. I think we'll finish up with one final question. Are there other membrane options instead of the one that you've mentioned here? Are there other membrane types, or there are other plates that can be used on the system?
That's a good question. So currently, this is the best. So the best membrane that we have found, and we've tested many, is a 10 kD regenerated cellulose filter plate. So we have looked at other types, including other vendors and things like APES, and it's interesting. There's really I would say that the regenerated cellulose is the best filter for this application. But it's not to say that there aren't other filters out there. It's just that because this consumable and set of consumables works so well, it's the one that we recommend currently. But we have had experiments with APES that suggest that other membranes are capable as well. But we would not recommend that. And so these are the consumables that we highly suggest to use.
Great. So that brings us to the end of our Q&A session. If you have any other questions, please don't hesitate to contact us. And at this time, we'll have the webinar. So thank you so much for joining us today. I'd like to invite you to visit our website and any previously recorded webinars we have on our automated systems for formulation development. And on behalf of Russell and myself, we'd like to thank you for attending, and we look forward to seeing you at future events. Have a great day.